detoxification mechanism जो discuss करणार आहे. I expect that from your side we will get some feedback, right? So you will have material available with you. I will show you some material. So one by one person they can explain, right? We don't expect anybody to have very serious understanding, but just we will see if people can explain it, right? Hey, this like a detoxification. This like a is detoxification. This is this page visible? Vijay, Vijay.
Yes, sir.
हा तुम्हाला हे डिटॉक्सिफिकेशनचे इमेज दिसते.
Yes, sir.
ओके, तर आपण जेव्हा when we say detoxification by the liver, so what things comes to your mind when you see this image? ही इमेज दिसते का तुम्हाला विजय?
Yes, sir. It is visible.
ओके, तर ही इमेज बघितल्यानंतर तुम्हाला डिटॉक्सिफिकेशन विषयी काय काय समजतंय की व्हॉट डिटॉक्सिफिकेशन इज रिलेटेड टू अँड व्हॉट मेसेज डज इज कन्वे डिटॉक्सिफिकेशन? Anyone else they can explain detoxification? Rohini, Rohini Kupse, am I audible? Okay, Gauresh.
Removal of toxic substance.
Okay. Okay, Gauresh, am I audible?
Hello, sir.
Rohini.
Removal of toxic substances from the living organism, and that is mainly carried out by the liver.
ओके, स्क्रीन दिसतंय का तुम्हाला? Is screen visible, Vijay?
Sir, it is not visible now.
Yes, it is visible there.
Yes, sir, it is not visible.
Now, is it visible? The detoxification by the liver?
Yes, sir, it is visible now.
Okay, Vijay, आपल्याकडं we have detoxification by liver, one topic, right? So we are not going in detail, but just by images we are going to discuss something, right? We don't want to go in detail. So what you can deduce from this figure image? What do you mean by detoxification?
Sir, whatever the harmful substances are present in bloodstream,
Mhm.
they are converted into less harmful substances and they are removed by liver.
Yes, good. So when we say detoxifications, means it is some harmful substances. For example, we call them as a toxins, right? So toxins we can remove it by the detoxification process. And what are this flow diagram is represented? So what we can say from this flow diagram? Toxins is written, right? Here it is written toxins, fat soluble. Later, phase one, P45 enzymes, then intermediates, reactive metabolites, then antioxidant protection, intermediates, then phase two conjugation, then waste, water soluble, and then elimination via bowel in the feces and via kidney in the urine. So, what do you understand by this figure, Vijay?
Sir, detoxification involves two phases.
Hmm?
In phase one, enzymes act.
Phase one enzymes P450 enzymes, right? The cytochrome P450 enzyme group is there, right? Okay.
Yes, sir.
Next.
There is an intermediate stage between phase one and phase two.
Okay, intermediate metabolite will be formed between phase one and phase two. Next.
Then after phase two, the waste, water soluble waste is eliminated via bowel or kidney.
Okay, that's it. What happened in phase two reaction?
Sorry, sir, that
Conjugation happens, right? Conjugation means joining two things together. That is called as a conjugation, right?
yes, sir.
नंतर गौरेश तुम्ही सांगा. Gauresh, am I audible?
Yes, sir.
Okay. Next thing is there, phase one and phase two is there. So which reactions can happen in the phase one reaction? So yeah, can you see this image? This way, one image we are showing it. Is it visible?
Yes, sir. Yes, sir.
Okay. So what can you say from this image?
In phase one reaction, fat soluble toxins are
Fat
used, sir.
soluble toxins. Okay.
Cytochrome P450 enzymes.
Okay.
In phase one reaction, there are three types. Five types: oxidation, reduction, hydrolysis, hydration, and dehalogenation. Five
Okay.
steps.
What we mean by oxidation?
Sir, and nutrients needed in phase one are vitamin B2, B3, B6, and B12,
Mm-hmm.
folic acid, glutathione,
Glutathione. Hmm.
flavonoids.
Okay. And when we say phase one reaction involve oxidation, reduction, hydrolysis, hydration, dehalogenation, right?
Yes, sir.
So what we mean by oxidation? If
Ah,
fat soluble toxins are there, they are undergoing oxidation. So what we mean by oxidation?
so with the help of oxida, they are oxidized to acid, sir.
Oxidation is the removal of electrons, right?
Sir, electron removal of electron.
Electron or some addition, sometime addition of oxygen or removal of this one hydrogen atom, right?
Yes, sir.
Okay, and reduction reaction.
Gaining of electrons.
Gain of electron, right, or gain of hydrogen atom. Hydrolysis reaction.
Removal of water.
Hydrolysis means with the help of water, we are adding some water in that hydrolysis reaction, right? Removal of water is a dehydration reaction, hydrolysis. And hydration,
Hydration. Addition of water.
okay, it is also involves the water addition. The dehalogenation reaction,
Remove of halogen.
halogen compounds is there, like chlorine, likewise compound are there, right? So the halogen atoms, iodine, chlorine, likewise
Chlorine, bromine,
chlorine next.
bromine, iodine.
Yes, yes. Chlorine, bromine, iodine. If you are removing it, we call it as a dehalogenation reaction, right?
Yes, sir.
So in phase one reaction, basically, we are doing oxidation, reduction, hydrolysis, hydration, and dehalogenation reaction. And any guess why this nutrient, vitamin B2, B3, B6, B12, folic acid, glutathione, flavonoids, why they are required?
Sir,
Why these vitamins will be required in the phase one reaction? Any idea?
to boost up the process.
Okay, so during the enzymatic reaction, these may be required, right? Okay, so this was about fat soluble toxins. Is the toxins which, if available in our body, and if they comes in contact with the fat, they get solubilized in the fats, right? So cytochrome P450 enzymes are there. And why there is a 450 written in front of enzyme P, cytochrome P450? Any guess?
Shape of enzyme, sir.
Cytochrome P450 is an enzyme, and 450 is a this absorption maximum for this enzyme, right? The enzymes it absorbs at the maximum at the 450 nanometer, right? Wavelength. It is related to wavelength.
Okay, sir.
Okay. Next, we have water soluble waste, right? So who else is there? So only two people are there. Okay. So water soluble waste are there. So what happened in the phase two? Vijay, you tell what happened in the water soluble waste to water soluble waste. Vijay.
Yes, sir. The water soluble waste is eliminated via urine, bile, or
Hmm?
stools.
Okay. Any guess why water soluble waste is being eliminated via urine, bile, and stool?
Because the bile pigments they give color to the feces.
Okay.
Hmm.
And because it is easily soluble in the water, and water is continuously removed from our body in the form of urine, right? And sometimes in form of the feces, also water is being removed. So along with that, this water soluble waste, it can be removed, right? Good. So what happened next? What happened in the water soluble waste removal process in the detoxification? Vijay.
Sir, the conjugation pathways.
हा फेज थ्रू मध्ये कंजुगेशन पाथवे असतं, राईट? अगोदरच्या फिगर मध्ये बघितलं होतं, तुम्ही सांगितलं होतं की फेज टू मध्ये कंजुगेशन पाथवे. तर कंजुगेशन मध्ये कोणत्या कोणत्या रिएक्शन आहेत? विच रिएक्शन्स आर देयर?
They are sulfation, glucuronidation,
ग्लुकोरोनायडेशन,
uh, glutathione
ग्लुटाथिओन
conjugation,
कंजुगेशन.
acetylation, amino acid conjugation, and methylation.
Okay. Any guess what is sulfate? What we mean by sulfation?
Means addition of sulfur.
Okay, sulfur addition may be there. Glucuronidation.
Sorry, sir, no idea.
Glucuronidyl group add होतो, right? Glutathione conjugation. Glutathione group conjugation म्हणजे काय? So conjugation means basically joining two molecules together, right?
Yes,
So
sir.
we have a water soluble molecule, and how we make water soluble waste go away? We uh, make them conjugated with some molecule like sulfur molecule or glucuronidal molecule or glutathione molecule. We conjugate our water soluble waste with these molecules, and once they are conjugated with them, they become water soluble, right?
Yes, sir.
Mostly in the phase two reaction. So glutathione conjugation, acetylation means addition of acetyl group. Amino acid conjugation means we are conjugating amino acid. Methylation means we are adding some methyl group to these compounds. So these are the different conjugation pathways. आणि कोणते न्यूट्रिएंट्स रिकॉर्ड असतात यामध्ये?
यामध्ये मिथिओनाइन, सिस्टीन,
हम्म,
मॅग्नेशियम,
हम्म,
glutathione, vitamin
हम्म.
B5, B12. Then
Okay,
there is vitamin C, glycine,
okay.
taurine, glutamine, folic acid, and choline.
Pulling. Okay, so this basically this image itself detect that. So intermediary metabolism. So once we have fat soluble toxins, we process them via oxidation, reduction, hydrolysis, hydration in the phase one reaction. Then they go into the phase two reaction, like this figure, right? So this is the toxins. They are undergoing in the phase one reaction, oxidation, reduction, hydration. Likewise, then intermediary metabolites form, and then they undergo the conjugation. And conjugation मध्ये काय होतं की हे जे ग्रुप्स जे आहे सल्फेशन, ग्लुकुरोनायडेशन, ग्लुटाथिओन, acetylation, amino acid conjugation, methylation, हे होऊन हे these become more water soluble, right? So they can be easily excreted in the form of urine, bile, and the stools. So the excretion is easier for this molecule. So you have understood this image, right? So let's see one more image if we have. So where these processes occur?
It happens in liver, sir.
Liver. Okay. So here one more image is there. Gauresh there?
Yes, sir.
Okay. Gauresh, can you see this image?
Yes, sir.
Okay. So can you tell what is happening in this image?
So non-end products, metabolites,
Non-metabolites. Okay.
and environmental toxins, bowel toxins are comes into liver.
Mm-hmm.
With the help of cytochrome P450 enzyme, they are activated into intermediates,
Mm-hmm.
and they are converted into with the conjugate pathways. They are converted into sulfation, glucuronidation, conjugation,
Mm-hmm.
and they excrete through the liver, sir.
Acha, exit to liver. Phase three excretion means maybe it's like excretion through urine or feces or bile, right?
Yes, sir.
Okay.
And,
So,
and bacteria and immune complexes are destroyed by converted into Kupffer cells,
okay,
and they are destroyed in the liver
okay,
only.
okay. So, in the liver, they may bacteria, they may get destroyed in the Kupffer cell, and they may produce some toxins, right? Toxins, and these toxins may be processed via cytochrome P450, right?
Yes, sir.
So you are getting idea how this pathway is working actually, right?
Yes, sir.
Okay, so let's see what we can see from this image. What we are, what you can say from this image. So we were describing bowel toxin, environmental toxin, non-invertebrate. So does this phase one and phase two pathway, does this produce only process only exogenous toxins, or does it process endogenous toxins also?
Yes, sir. Endogenous also, sir.
Okay, let's see here. Can you identify some? Okay. Uh, tell us what is happening in this figure.
Sir.
Can you explain what is happening in this image? Is it visible image?
Yes, sir.
Okay.
Drug metabolism is happening, sir.
Drug metabolism actually. So we are seeing some diet, infection, and xenobiotic substances is there. So what do you mean by xenobiotics?
Don't know, sir.
Xenobiotics. Vijay, xenobiotics.
Sir, the toxin which are produced in the diet metabolism.
Okay. Okay. Genome is basically other than our body, right? So genobiotic, if you see here, genobiotic, foreign chemicals in the body, chemical including drug, foodstuff normally located outside the body. So these are the compounds which are not located in our body, right? So other environmental toxins and all those things, this comes under the genobiotics. Next, one more is there, injury. Some injury is happening to our body or chemical exposure is happening. So steroid metabolism is there. So is steroid exogenous or endogenous substances? विजय स्टिरॉइड जे आहेत आपल्या बॉडीमध्ये ते एंडोजिनस सबस्टन्सेस आहे की एक्झोजेनस सबस्टन्सेस आहेत
Sir, they are endogenous substances. Endogenous substances.
Endogenous, right? Testosterone, estrogen, these are being produced in the animals, right? So we can catabolize this exogenous substance via this cytochrome P3A4. So what do we mean by Vijay? What do we mean by CYP? Capital C.
Cytochrome P.
That is cytochrome.
सायटोक्रोम पी राईट सायटोक्रोम पी आणि हे जे दुसरे काय त्याचे सबटाईप आहे जसं सायटोक्रोम P1, सायटोक्रोम P2, सायटोक्रोम P3, P4. Likewise, different different types are there cytochromes, and of these cytochromes, this A, B, C, D, these are the subtype, right? Subtype. So likewise, we are the and म्हणजे सायटोक्रोम टी एक टाइप झाला फॅमिली झाली सायटोक्रोम P3 आणि सायटोक्रोम 3A is a subfamily and then cytochrome 3A4 is a isotype of this A family subfamily, right? Are you getting this one?
Yes, sir.
Okay, means cytochrome P3 is a family, cytochrome P is a family, P3 is a one family, then cytochrome 3A, B, C, like cytochrome 3A, 3B, 3C. Likewise, these are subfamily, and then cytochrome 3A, 1, 2, 3, A, A1, A2, A3. These are like subtypes, right? Isotypes. So these are different types of subtypes of enzymes present in the liver. Each will have different functions of either undertaking the glucuronidation, like hydration, oxidation. Likewise, these functions are there, right? Cytochrome. So these enzymes is basically will be helping in the fat metabolism in the liver and the drug desactivation and procarcinogen desactivation. So when we give a drug to animal. So what happened to this drug? Gauresh?
Sir, drug metabolism.
ड्रग मेटाबॉलिझम होतं तर ड्रग आपण सपोज एखादं ड्रग दिलं की सपोज नॉन स्टेरॉइडल अँटी इन्फ्लेमेटरी ड्रग जे आहे तर बरेच ड्रग जे असतात ते लिव्हरला अफेक्ट करतात कशामुळे अफेक्ट करतात ते ड्रग्स ऍज अ साइड इफेक्ट समटाइम्स सम लिव्हर प्रॉब्लेम मे हॅपन वेन वी टेक मोर ड्रग्स व्हाय इट हॅपन्स
Excessive drugs can destroy the liver, sir.
Yes, excessive drugs can destroy the liver. But why they can destroy the liver?
Liver can't accept that much drugs, sir.
Okay. Because sometimes drugs can be toxic. Once their effect, beneficial effect is done at the target site, at the other side, these drugs can be toxic for the body, right? So body treat them as a toxins and body tries to get removed. The body tries to get rid of them, right? With the help of this phase 1 reaction, phase 2 reaction in the liver, it tries to get rid of this reaction. And if load of this one increases, then it may damage the liver, right? So sometimes what happens is that once you take this drug, these drugs can get inactivated and it can be easily get removed in the feces or in the urine. And the other time, sometime what happens, some drugs are there, they will come here to the liver, and with the help of the liver enzyme, they will be converted into the more active form of the drug, which could be a more toxic. That also sometimes happen, and which may damage the liver or sometimes kidneys. So these things can happen, right? Are you getting role of this cytochrome P detoxification pathway in the liver?
Yes, sir.
Drug metabolism, right? So likewise, drug metabolism pathway. So let's see one image here, one more image. Okay, same thing. What can you understand from this? XME means xenobiotic metabolizing enzyme, right? So Vijay, what we understand from this image here?
Sir, there is a metabolic activation.
Hmm?
What
No, no.
the
Hmm. Okay.
whatever the environmental pollutants like drugs, foodstuffs, heavy metals.
Okay. These environmental pollutant, drugs, food stuff, heavy metals. These are what we are calling it as a whether they are present in our body,
No,
animal
sir.
body or human body. No. So
No.
what we call them
Xenobiotics.
xenobiotics, right? Because they are not
Yes.
part of us. Okay. Next.
In the phase first, cytochrome
Phase.
P1234,
These are cytochrome phenol in the phase one reaction. Okay.
they act they act on xenobiotics,
They act on xenobiotics. Okay.
and there are functionalization reactions.
ओके फंक्शनलायझेशन रिएक्शन ही तो कोणत्या कोणत्या फंक्शनलायझेशन रिएक्शन्स होतात त्यामध्ये फेज वन रिएक्शन मध्ये ऑक्सिडेशन रिडक्शन हायड्रेशन हायड्रोलायसिस
And
likewise
dehalogenation. Yes.
reaction
Dehalogenation.
dehalogenation reaction good so this reaction happen in the phase one reaction, right? So this is a functionalization reaction. Next, what happened?
Then there is a metabolic activation.
uh, metabolic activation happens. Okay, so metabolic activation मध्ये काय होतं?
The reactive oxygenated metabolites, they can be carcinogenic and mutagenic through binding to DNA or protein.
Okay, reactive oxygenated metabolism which can be carcinogenic and mutagenic through binding to the protein. Okay,
After that, there is oxidative stress, mutations occur,
okay,
toxicity, and cancer.
okay. So under the phase one reaction, some environmental pollutants are there, drugs are there, foodstuffs are there, or heavy metals are there. Okay, they are undergoing phase one reaction, and they are converted into the metabolic activation. Functionalization of reaction is happening. Oxidation, reduction, dehalogenation, hydration, hydrolysis. And after this reaction, this reactive oxygen metabolite can be produced. It can be a carcinogenic, means it can cause the cancer. It can be mutagenic, means it can cause mutation. And because of this mutagenic or the carcinogenic reactive oxygen species, we call as a ROS. There can be mutation, toxicity, or the cancer. Okay. Uh,
Yes, sir.
so, so what we can do to avoid that? Phase one reaction, नंतर reactive oxygen species तयार झाले, carcinogenic and mutagenic substances have been produced. So next, what we can do with those substances? There is a phase two reaction, right?
Yes, sir.
Gauresh,
Yes, sir. Phase two reaction is there.
phase two reaction is there, right? So we can take these metabolites under the conjugation reaction, right?
Like
We
sulfation,
can
glutathione
conjugation.
conjugation, acetylation,
Acetylation, then
sulfation.
sulfation, glucuronidation, these are the reactions which can be Okay. Am I audible? Okay. Vijay,
Yes, sir.
हा याच्यामध्ये आपल्याकडे जेनेटिक विजिबल.
Sir, your slide not visible.
Okay, 1 minute. Now is it visible?
Yes, sir, it is visible now.
Okay. So we have this exposure to genobiotics, right? So there is term called as a genetic polymorphism. So sometimes we say that some people, some animal, they are responding to some particular drug, and some animal they are not responding to particular drugs. So why this is happening? So some animal they are showing some side effect, and other animal they are not showing side effect. Or even if we take a particular drug, some people have side effect to that drug, and other people don't have side effect to that drug. One of the mechanism, what could be one of the mechanism based on this image? Gauresh,
Sir, don't know, sir.
Gauresh, Vijay, are you getting my point?
Yes, sir.
So we have two people, right? One people has some pain. Both people have pain. They went to the market and they have taken some anti-inflammatory tablets. And these anti-inflammatory tablets, both of people taken, but one have severe reaction and other doesn't have severe reaction as a side effect. So why these things can be happening?
Because of gene polymorphism.
Genetic polymorphism, right? So genetic polymorphism is some this cytochrome P enzyme is there, right? Cytochrome P1, 2, 3, 4 enzyme we have. Okay, Gauresh,
Yes, sir.
am I
Yes, sir.
audible? Okay, so already we have seen phase one and phase two reaction little bit. Can you read this phase one reaction for us? Means phase one enzymes are there, right? Phase one enzymes are there, phase two enzymes are there. When we say phase one reaction, phase two. So cytochrome
Hydrolysis,
P450 was one of the enzymes. So what are the other? Hmm?
hydroxylases, reductases,
Okay,
aldoketoreductases,
aldoketoreductases. Next.
flavin containing monooxygenases,
Okay,
lipooxygenases,
okay,
cyclooxygenases, peroxidases, epoxygenases, oxidases,
okay,
amine oxidases, dioxygenases, NAD
okay,
dependent, NAD independent alcohol dehydrogenases,
okay,
NAD dependent and NADP dependent aldehyde dehydrogenases,
okay.
NAD dependent and NADP dependent steroid dehydrogenases,
Okay.
carboxylases,
Okay.
glycosylases, glucuronidases.
Glucuronidase, hydrolysis, esterases. So,
Yes.
Gaurish, tell me, we have seen cytochrome P450.
Yes.
So, is this the only enzyme involved in this detoxification processes, or other enzymes also involved?
Other enzymes also involved.
Other enzymes also involved, right? Previously, we have showed,
Major portion taken by cytochrome P450s.
yes, major portion is done by cytochrome P450, like cytochrome P1, P2, P3, P4 enzymes are there. But there are enzymes what we have you have read right now, that is hydroxylases, reductases, peroxidases, oxidases, glycosylases. These are enzymes also modifying some toxins, right?
Yes, sir.
Okay. So Vijay, you tell phase 2 enzymes. Phase 2 enzymes कोणते कोणते आहेत आपल्याकडे?
Sir, uridine diphosphate glucuronosyl transferases, then glutathione.
Glucuronil transferase. It means that this enzyme, okay, glutathione is transferase. So it is transferring glutathione. Okay, next.
Sulfotransferases, epoxydases.
So what this enzyme is doing? Sulfotransferase, it is transferring sulfur
Yes,
group,
sir.
right? Okay, epoxidases. Next.
Acyl transferases, then acetyl transferases,
Okay,
methyl transferases,
okay,
transaminases.
okay.
Yes.
Transaminases. Okay, so basically these are different enzymes. So basically, firstly, we have seen the short list of these enzymes, and now it is a long list of enzymes. But basically, these are the enzymes which are involved in the detoxification process, right? Next one more have established being responsible, hypokalemia. So we will try to get it. Okay, some phenytoin and warfarin oxidation defectivity is there. So this enzyme defect can be involved, right? Cytochrome P2, capital C9. So you don't have to remember all these names in detail. Cytochrome P2, C9. But this is one of the example that if this CYP2, C9, if this gene is defective, then this phenytoin and warfarin oxidation defect can be there. Okay. Okay. One more here. Cytochrome P1. This has a two families. It has a 3 number of gene, and its function is a metabolism of eicosanoid, genobiotics. In addition, CYP1, A2 metabolites, melatonin, estrogen, urocorpanogen, and 24 enzymes. Cytochrome P5, number of subfamily is 1, number of genes is 1, and substrate is a thromboxane A2 synthetase. Okay, so Gauresh,
Yes, sir.
so what can you say from this table? What this table represents?
Cytochrome has different types like
Cytochrome has a different types. Okay,
P2, P3, like up to P27.
okay. Actually, P51 till 51. Okay, next.
Okay. And it's showing their substrate or functions.
Different substrate functions are there. Like if we have this function, like this thromboxane A2 synthesis is there, synthase is there. If it has to be get activated, then cytochrome P5 is involved, right?
Yes, sir.
And if it is a steroidogenesis related to steroidogenesis, this is a cytochrome
P11.
P11 is involved, right? Okay, so and these are the number of genes. There is a 3 gene involved for the steroidogenesis cytochrome P11 and 2 subfamilies likewise. Okay, let's see if any more interesting image is there. So sometimes some substances are there. They are causing cancer. So can you identify why they are causing cancer? So this is a polychlorinated biphenyl compound, right? PCB means polychlorinated biphenyl compound, and PAH. Some pH substance is there. So basically, polyphenolated compound, right? Biphenyl compound. So why they can cause cancer? Uh, Vijay, can you tell why they could be causing cancer? Vijay?
Yes, sir.
We have xenobiotics, right? For example, PAH or PEH, AH, including this PCB. That is, PCB is a
Yes.
fluorochlorinated biphenyl compound, right? Biomolecules. These are
Yes,
and some indigenous effector. So why they could be causing cancer? Can you say from this image?
sir. Because they damage DNA and there
Okay.
is mutation, so cancer happens.
So cancer happens. So is this detoxification mechanism useful or not?
So it is useful somewhat.
Somewhat useful means most of time they try to detoxify. They try to prevent these chemicals which cause DNA damage. But in other time, if these compounds, likewise this PCB compound is there, they may be get activated at the activity to more carcinogenic compound, and then they may cause cancer, right?
Yes.
So it is a both way functions can be there, right? Okay, so that's it for today's discussion, right? So basically, we want to know what is cytochrome and what is detoxification microorganism. Are you getting the point, Doresh Vijay?
Yes, sir.
Yes, sir. We are getting.
You are getting point, right? We have phase one metabolism, phase two metabolism, and then we have this pathway like oxidation, reduction, hydration, dehydration, desulfonation, halogenation. आणि फेज टू मध्ये कोणते कोणते पाथवे बघितले आपण? विजय,
Yes, sir.
फेज टू मध्ये कोणते बघितले आपण? पाथवे विच पाथवे डिड वी सॉ इन द फेज टू रिएक्शन?
In phase two, there is
Conjugate.
acetylation, methylation,
Acetylation, methylation. Okay, next
sulfation.
sulfation. Next
Glucuronidation.
Gauresh,
Glucuronidation.
glucuronidation. These reactions we saw, right? Glucuronidation. These
Glutathione candidate.
things we saw, right? Glutathione, yeah, glutathione transfer reaction also we saw. Okay, so basically we have this water soluble reaction, the fat soluble in the phase one reaction. Water soluble making reaction is a conjugation reaction, and then they can be excreted via the kidney or the feces, right? Okay, so that's it for today, right, Gauresh Vijay? So next time we will try to discuss in detail again, likewise one day, right? So likewise, you will be discussing.